Chapter 8 Cell, the unit of life. Point 6 Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Pallard, 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Here S, Svedberg's unit, stands for the sedimentation coefficient. It indirectly is a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. 8.5.7 Cytoskeleton. An elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and Flagella. Cilia, singular, cilium, and flagella, singular, flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules, and a pair of centrally located microtubules. Such an arrangement of axonal microtubules is referred to as the 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. 8.5.9 Centrosome and centrioles. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cells.